Hello, welcome to Biology, the scientific study of life. Antimicrobial proteins. The antimicrobial action of lysosome. Other antimicrobial proteins include about 30 serum proteins that make up the complement system. In the absence of infection, these proteins are inactive substances on the surface of many microbes. However, can trigger a cascade of steps that activate the complement system, leading to lysis, bursting of the invading cells. Certain complement proteins also help trigger inflammation or play a role in acquired defense. Two types of interferon, alpha and beta, provide innate defense against viral infections. These proteins are secreted by a virus infected body cells and induce neighboring uninfected cells to produce other substances that inhibit viral reproduction. In this way, interferons limit the cell-to-cell spread of viruses in the body, helping control viral infections such as colds and influenza. This innate defense mechanism is not virus-specific. Interferons produced in response to one virus may also confer short-term resistance to unrelated viruses. Certain lymphocytes secrete a third type of interferon, U or gamma, that helps activate macrophages, enhancing their phagocytic ability. Interferons can now be mass produced by recombinant DNA technology and are being tested for the treatment of viral infections and cancer. Yet another group of antimicrobial proteins, fittingly named defenses, are secreted by activate macrophages. These small proteins damage broad groups of pathogens by virus mechanisms without harming body cells. An inflammatory response. Damage to tissue by physical injury or the injury of pathogens leads to release of numerous chemical signals that trigger a localized inflammatory response. One of the most active chemicals is histamine, which is stored in mast cells found in connective tissues. When injured mast cells release their histamine, triggering dilation and increased permeability of nearby capillaries. Activated macrophages and other cells discharge additional signals such as prostaglandins that further promote blood flow to the injured site. The resulting increased local blood supply causes the redness and heat typical of inflammation. From the Latin inflammare, to set on fire. The blood engorged capillaries leak fluid into neighboring tissues, causing swelling, another sign of local inflammation. Although heat and swelling are uncomfortable sensations, the enhanced blood flow and vessel permeability that cause them are critical to innate defense. These vascular changes help deliver antimicrobial proteins and clotting elements to the injured area. Several activated complement proteins, for example, promote the release of histamine to attract phagocytes to the site. Blood clotting begins to repair process and helps block the spread of microbes to other parts of the body. In addition, increased local blood flow and vessel permeability allow more neutrophils and monocyte macrophages to move from the blood into injured tissues. Small proteins called chemokines direct the migration of these phagocytes and signal them to increase production of microbe-killing compounds. Chemokines are secreted by many cell types, including blood vessel, endothelial cells near a site of injury or infection, as shown in this figure. A minor injury causes local inflammation, but the body may also mount a systemic widespread response to severe tissue damage or infection. Injured cells often put out a call for reinforcement, secreting chemicals that stimulate the release of additional neutrophils from the bone marrow in a severe infection, such as meningitis or appendicitis. The, the number of white blood cells in the blood may increase several fold within a few hours of the initial inflammatory events. Another systematic response to infection is fever. Fever may occur when certain toxins produced by pathogens and substances released by activated macrophages set the body's thermostat at a higher temperature. A very high fever is dangerous, but moderate fever may facilitate phagocytosis, and by speeding up body reactions, hasten the repair of tissues. Certain bacterial infections can induce or overwhelming systematic inflammatory response, leading to a condition known as specific shock, characterized by very high fever and low blood pressure. Septic shock is a common cause of death in hospital critical care units. Clearly, local inflammation is essential for healing, but systematic inflammation can be devastating. Back to major events in the local inflammatory response. Picture one chemical signals released by activated macrophages and mast cells at the injured site cause nearby capillaries to widen and become more permeable, as shown in this figure. Two fluid antimicrobial proteins and clotting elements move from the blood of the site clotting begins. Three chemokines released by various kinds of cells attract more phagocytic cells from the blood to the injury site. For neutrophils and macrophages, phagocytous pathogens and cell debris at the site and the tissue heals. Notice that. Natural killer cells. We wrap up our discussion of vertebrate innate defenses with natural killer NK cells. NK cells patrol the body and attack virus infected body cells and cancer cells. Surface receptors on an NK cell recognize general features on the surface of its targets. Once it is attacked or attached to a virus infected cell or cancer cell, the NK cell releases chemicals that lead to the death of the stricken cell by apoptosis or programmed cell death. Although defense provided by NK cells is not 100% effective, viral infections and cancer would occur much more often without these innate sentinels of the body. Invertebrate immune mechanisms. Before we move on to examine acquired immunity in vertebrate, we should know that invertebrates also have highly effective innate defenses. For example, sea stars possess amoeboid cells that ingest foreign matter by phagocytosis and secret molecules that enhance the animal's defensive response. Recent studies of the fruit fly Drosophila Melangastor also have revealed striking parallels between insect defenses and vertebrate innate defenses. The exoskeleton of insects, like the skin and mucous membranes of vertebrates, provides an external barrier that can prevent entry of intruders. If an insect's skeleton is damaged, pathogens that enter the insect's body must face several internal innate defenses. The insect equivalent to blood, the hemolymph, contains circulating cells called hemocytes. Some hemocytes ingest bacteria and other foreign substances by phagocytosis, while other hemocytes from a cellular capsule around a large parasite. The presence of pathogens signals still other hemocytes to make and secrete various antimicrobial peptides that bind to their pathogens targets, leading to death of the pathogens. The internal signaling pathways that trigger hemocytes to produce antimicrobial peptides are comparable to those that activate vertebrate macrophages. In addition, certain hemocytes contain the enzyme phenoloxidase. Once activated, this enzyme converts phenols to reactive compounds that link together into large aggregates. These are deposited around parasites and wounded tissue, helping to prevent the spread of parasites beyond the affected area. Activation of phenoluxidase in insects occurs by a cascade of steps similar to those that activate vertebrate complement proteins. Recent research indicates that invertebrates lack cells analogous to lymphocytes, that white blood cells responsible for acquire specific immunity in vertebrates. But even though they depend on innate, non-specific mechanisms, certain invertebrate defenses do exhibit some features characteristic of acquired immunity. For instance, acquired immunity usually is directed against self cells, not normal body self cells. The ability to distinguish self from non-self is seen in the least derived animal lineage. The sponges, if cells from two sponges of the same species are mixed, the cells from each sponge sort themselves and reaggregate, excluding cells from the other individual. Another hallmark of acquired immunity is immunological memory of or the ability to respond more quickly to a particular invader or foreign tissue the second time it is encountered. Earthworms exhibit something like this. Phagocytic cells in a worm attack a second graft from the same donor worm much more rapidly than the first graft. Most invertebrates, however, exhibit no such immunological memory. In acquired immunity, lymphocytes provide specific defenses against infection. While invading pathogens are under assault by vertebrates, innate defenses, the intruders inevitably come into contact with lymphocytes, the key cells of acquired immunity, the body's second major kind of defense. Direct contact with microbes and signals from active innate defenses cause lymphocytes to join the fight. For example, as macrophages and dendritic cells phagocytose microbes, the phagocytes begin to secrete cytokines, proteins that help activate lymphocytes and other cells of immune system. This is just one example of how innate and acquired defenses interact. Any foreign molecule that is specifically recognized by lymphocytes and elicits a response from them is called an antigen. Most antigens are large molecules, either proteins or polysaccharides. Some antigens, such as toxins secreted by bacteria, are dissolved in extracellular fluid, but many protrude from the surface of pathogens or transplanted cells. A lymphocyte actually recognizes and binds to just a small accessible portion of an antigen called an epitope or antigenetic determinants. A single antigen usually has several different epitopes, each capable of inducing a response from lymphocytes that recognize that epitope antibodies, which are secreted by certain lymphocytes in response of antigens, likewise bind to specific antitopes. In this section, okay, more about antigen recognition by lymphocytes. The vertebrate body is populated by two main types of lymphocytes: beta lymphocytes, B cells, and T lymphocytes, T cells. Both types of lymphocytes circulate through the blood and lymph and are concentrated in the spleen, lymph nodes, and other lymphoid tissues. B cells and T cells recognize antigens by means of antigen-specific receptors embedded in their plasma membranes. A single B or T cell bears about 100,000 of these antigen receptors, and all the receptors on a single cell are identical. That is, they are recognized the same epitope. In other words, each lymphocyte displays a specificity or a particular epitope on an antigen and defends against that antigen or a small set of closely related antigens. As receptors for antigens, each B cell receptor for an antigen is a U-shaped molecule consisting of four polypeptide chains, two identical heavy chains, and two identical light chains linked by disulfide bridges, a region in the tail portion of the molecule, the transmembrane region, anchors the receptor in the cell's plasma membrane, and short region at the end of tail extends into cytoplasm at the tips of the U are the light and heavy chain variable V regions. Notice this figure. So named because their amino acid sequences vary extensively from one P cell to another. The remainder of the molecule is made up of the constant C regions, whose amino acid sequences vary little from cell to cell. As shown in this figure, each B cell receptor has two identical antigen binding sites. The unique contour of each binding site is formed from part of a heavy chain V region and part of light chain V region. The interaction between an antigen binding site and its corresponding antigen is stabilized by multiple non-covalent bonds between chemical groups on the respective molecules. The antigens bound by B cell receptors in this way include molecules that are on the surface of or are released from two types of infectious agents. In other words, a B cell recognizes an intact antigen in its native state. Secreted antibodies or immunoglobulins are structurally similar to B cell receptors, but they lack the transmembrane regions that encode receptors in the plasma membrane. Because of this structural similarity, B cell receptors are often called membrane antibodies or membrane immunoglobulins. Immunoglobules. Cell's plasma membrane at the outer tip of the molecule, the alpha A and B chain variable V regions form a single antigen binding site. The remainder of the molecule is made up of the constant C regions. T cell receptors recognize and bind with antigen just as specifically as B cell receptors. However, While the receptors on B cells recognize intact antigens, the receptors on T cells recognize small fragments of antigens that are bound to normal cell surface proteins called MHC molecules. MHC molecules are so named because they are encoded by a family of genes called the major histocompatibility complex MHC. As a newly synthesized MHC molecule is transported to the plasma membrane, it binds with a fragment of a protein's peptide antigen. Antigen presentation, a nearby T cell can detect the antigen fragment thus displayed on the cell surface. There are two ways in which foreign antigens can end up inside cells of the body depending on their source. These peptide antigens are handled by a different class of MHC molecule and recognized by a particular subgroup of T cells. Class number one. Class 1 MHC molecules found in almost all nucleotide cells of body-bound peptides derived from foreign antigens that have been synthesized within the cell. Any body cell that becomes infected or cancers can display such peptide antigens by virtue of its class 1 MHC or MHC molecules. Class 1 MHC molecules displaying bound peptide antigens are recognized by a subgroup of T cells called cytotoxic T cells, as shown this figure. Et voilà. Class number 2. Two MHC molecules, these are made by just a few cell types, mainly dendritic cells, macrophages, and B cells. In these cells, class 2 MHC molecules bind peptides derived from foreign materials that have been internalized and fragmented through phagocytosis or endocytosis. Dendritic cells, macrophages, and B cells are known as antigen-presenting cells because of their key role in displaying such internalized antigens, or another subgroup of T cells called helper T cells, as shown this figure. Each vertebrate subspecies possesses numerous different alleles for each class 1 and class 2 MHC gene. MHC proteins are the most polymorphic known because of the large number of different MHC alleles in the human population. Most of us are heterozygous for every one of our MHC genes and produce a broad array of MHC molecules. Collectively, these molecules are capable of binding and presenting a large number of peptide antigens. Moreover, any two people except identical twins are very unlikely to have exactly the same set of MHC molecules. Thus, the MHC provides a biochemical fingerprint unique to virtually every individual that marks body cells as self. In fact, the discovery of the MHC occurred in the process of studying the phenomena of skin graft rejection and acceptance. Thanks for listening.